Hello and welcome to another episode of Momento Malum today. In this episode, we'll be talking about pea genetics. Let's get started. After the pea was domesticated, it was further developed by people into multiple domesticated subspecies. This includes subspecies arvensis, or the field pea, subspecies sativum, or the seed pea, and the subspecies hortensis, or the garden pea, the latter of which has edible pods. For the longest time, the analysis of genetic diversity was based on pedigree data and morphological traits, with a few number of molecular markers. Due to the high variability of these pea populations in the face of different environments, the adequate differences between different cultivars could not be measured. In more recent years, various types of genetic markers, such as RFLP, RAPD, AFLP, and SSR, have been discovered. SSRs have already been used extensively to assist genetic diversity in peas, soybeans, corn, cabbage, rice, and barley, as well as wheat. In peas, SSR markers were used to assess the genetic diversity of another population of peas and the overall evolution of this genus. It was also used to map the genome of the pea to find useful locations within the genome that have disease resistance. As a consequence of the improvements in more modern cultivars, with improvements in yield and disease resistance, there's been an extensive loss of land races, which is where most of the variability of the different populations dwell, and historically were the source of the different variations that led to the improvement in modern cultivars. The researcher Dr. Cupic of the University of Zagreb, Croatia, hopes to utilize the various morphological data, genetic markers, and pedigree data to determine the differences between two populations of both field pea and seed pea. Of the two subspecies being tested, there's a total of 18 pea cultivars evaluated, 11 field pea varieties, and 7 seed pea varieties. Of the 18 varieties in total, 3 of these cultivars are land races, while the rest are modern-day cultivars. These cultivars were tested using pedigree data, morphological traits, and SRR genetic markers, alongside various types of genetic software algorithms. Hello, I just want to let you know, if you want to fund this channel, you can do so on the platforms seen in the description below. The cultivars and graphs indicating the results will be showcased in the last 3 slides. Based on the pedigree data alone, the genetic difference between cultivars was on average 0.95 within the seed pea group and 0.96 on average between different cultivars in the field pea group. The average genetic distance between both groups was 0.99. This indicates that despite the loss of many of these land races, there's still a decent amount of genetic variation between individual cultivars using pedigree data. Using morphological traits, the average distance between different cultivars within the field pea and seed pea groups respectively were 0.54 and 0.52, indicating there's a lot fewer physical features that separate the cultivars based on physical features. Again, in terms of physical traits, the difference between both groups was 0.71. Using this morphological analysis, the researchers were able to divvy up the 18 cultivars into 3 different clusters. One consisted only of field pea cultivars, one consisted of a mixture of field pea and seed pea cultivars, and the third one was only seed pea cultivars. The genetic data was closer to the morphological data than it was the pedigree data. The average genetic difference between all 18 cultivars was 0.59. Within the field pea group, the average distance between different cultivars was around 0.53, and within the seed pea group is 0.49, and the average distance between both groups was 0.69. This indicates that, through the analysis of genetics, that there's less genetic variability than the pedigree would suggest, and the different cultivars are more closely related than previously suspected. Within the seed pea group, 2 distinct genetic clusters formed, one containing 6 cultivars and the other containing 3. The loss of genetic diversity is very likely because of the fixation of traits. As breeding happens, various traits become fixed through the breeding process, due to their continuous utility within the breeding program. And indeed, there's a high fixation index for this population, a 0.995 fixation index, with a minimum number of heterozygous individuals, which also makes sense due to the fact that it's a predominantly self-pollinating species. The researchers concluded that the amount of genetic diversity previously established using pedigree data was overestimated. However, it's still high enough to suggest that the genetic diversity in the gene pool of the common pea is still enough to create new variation from crossbreeding different cultivars. Evidence suggests that crossbreeding between the seed pea and field pea populations, as well as the revitalization of the old land races, and their subsequent incorporation into the gene pool, will help improve the commercial gene pool of common pea cultivars. And that about covers everything. Thank you for watching.